Hello, Bio 104 students. I thought it important to give you an additional very short lecture on the fossil record. So these are the topics that we'll be covering today in this abbreviated lecture on the fossil record. Macroevolution, remember we were categorizing evolutionary biology into microevolution, the study of individuals and populations, with macroevolution, which is interested in larger patterns in the evolutionary history of life on Earth. That is, phylogenetics in the fossil record provide the main lines of evidence for studying macroevolution. Now, the fossil record in its totality is called the Phanerozoic. That is the time on Earth where life, complex life, is recorded in the fossil record. And the Phanerozoic is broken into three periods: the Paleozoic, the Mesozoic, and the Cenozoic. And what demarks those three time periods are periods of the onset of the Paleozoic, the appearance of complex organisms in the fossil record in the Cambrian period. The end of the Paleozoic is characterized by a dramatic mass extinction event, the end Permian event. And the end of the Mesozoic is characterized by another dramatic extinction event, the Cretaceous Paleogene extinction event, which you may be familiar with because that was the time period where dinosaurs go extinct and where modern birds and placental mammals start appearing in the fossil record after the Cretaceous Paleogene mass extinction event. Now, the Phanerozoic is a time period comprising about 540 million years ago to the present day. And this is where animal and plant life becomes abundant in the fossil record. Now, why this is interesting is to consider that the Earth itself is about 4.5 billion years old. So life on Earth, as recorded in the fossil record, occupies a very small time slice of the total history of Earth. So this diagram here on your left is showing the massive amount of time in the Precambrian, which is the area prior to the Phanerozoic. And then we can blow up the Phanerozoic in the middle category here, showing you the three time periods of the Phanerozoic: the Paleozoic, the Mesozoic, and the Cenozoic. And then on the right, what we have is the Cenozoic blown up. So we're scaling on the far right from about 65 million years to the present, to your far left going back 4.6 billion years to the present. Now, how do we date rocks and fossils? Well, one approach that we talked about in lecture was biostratigraphy. Remember, we're matching up fossil species that are found in rock formations in different parts of the world. So we could correlate, say, rocks in Montana of Western North America, we would infer are the same age of rocks in Mongolia based on the fossils that are encased in those rock formations. Now there, we're only getting relative ages. We could say, okay, the ages of these rocks in Montana and China or Mongolia may be the same age. However, we're not getting an absolute age on what those rocks. One way is to look at isotopic ratios in the rocks of which the fossils are encased in, or rocks that are directly above or below the formations that contain the fossils. So many of you may have heard of carbon dating. Well, carbon dating is not really useful. It's not useful for fossils. Carbon dating is pretty much used for human archaeological studies where we actually have artifacts of organic materials. And we're looking at a half-life of about 5,700 years, and carbon dating is only useful on the scale of 60,000 to about 100 years ago. So clearly too young to be valuable for dating rocks that contain fossils. The most common isotopes that are used in dating rocks that are of interest to the fossil record are uranium-235 and potassium-argon isotopic ratios. And you can see here that they provide useful dating ranges for rocks that are on the scale of millions, tens of millions, if not hundreds of millions years old. Okay, now with fossils, the structural details that are encaptured can be amazing. So these are a couple of fossils preserved in amber. We have a 90 million year old fossil mushroom here. And I believe the bottom left is a 25 million year old fly that is, looks like a hymenopterum that is encased in amber. And the structural details provide really important insight into where these fossil taxa go in the tree of life. So for example, probably the most famous vertebrate fossil, Archaeopteryx, the very early bird lineage that is found in rocks in Germany. And what we see that what's interesting about Archaeopteryx, while it is closely related to birds, it has characteristics that birds don't have, such as having teeth, fingered hands, as well as a long, straight, bony tail. Now, there's an apocryphal story that O.C. Marsh, the founder of the Peabody Museum, had an opportunity to buy this fossil in the mid-19th century, but came down to haggling over something on the order of $300 in the mid-19th century, and walked away from purchasing the fossil. And so this iconic fossil is not at Yale, but is at a museum in Berlin. Now, sometimes you can get a lot of really interesting information from fossils, such as color. So here's a really important study on a Chinese non-avian dinosaur where feathers are preserved in the fossil. And because the pigments of the feathers are not technically fossilized, meaning the pigments are still there that were in the living organism, a study that involved Professor Richard Prum here at Yale EEB and Derek Briggs, Earth and Planetary Sciences here at Yale, were able to extract the pigments from these fossils and reconstruct the color of this feathered non-avian dinosaur. Now, with regard to biotic assemblages, that by looking at the organisms that are in the same fossil formation, scientists can reconstruct what assemblages looked like in the past. Here is a really interesting attempt to do that. This is from the wonderful mural in the Great Hall of the Peabody Museum, which is now closed, undergoing renovation, that was an iconic representation of life through the age of dinosaurs all the way through the end Cretaceous Paleogene mass extinction event. But the idea here is that even with this mural in the Peabody Great Hall, the representation of the animals and plants is not fanciful. These actually represent occurrences of these lineages in the fossil record. Now, we can also think about the origin and dominance of groups in the past based on the fossil record. So one thing we're going to talk about when we study plants is what Darwin called the abominable mystery of flowering plants. So flowering plants completely dominate living plant diversity today. There's about 250,000 species of flowering plants. And the question becomes, what in the evolutionary history did we see a turn of flowering plants dominating plant biodiversity? So what we're looking at here on the y-axis would be the fraction that these different phylogenetic lineages of plants are classified in the fossil record, right? So we could say the first 20% or so of all the plant fossils that we find in the Jurassic time period are conifers, pine trees. And we just go ahead and add up all the way to, we get 100% of the fossil record as represented. So if you see, if we go back to the Jurassic, there are no flowering plants. Flowering plants, the name are angiosperms. There are no angiosperms in the fossil record. But what we see is that as we go ahead and study the fossil record through time coming into the Cretaceous, and the Cretaceous being the last part of the Mesozoic, that what we come to is as we're just approached the middle part of the Cretaceous, what we see in the fossil record is a dramatic and very rapid rise of diversity and dominance of angiosperms in the fossil record relative to all of these other plant lineages. So in a very short time period, angiosperms go from not being present in the fossil record at all to dominating the fossil record by 80% or more, going through to the present day. So without this information, we wouldn't really have an understanding of what these previous biotic assemblages look like with regard to representation of these major plant groups. And we can see this dramatic appearance and dominating diversity of the angiosperms. Okay, also by compiling the information from the fossil record, so think of large databases where one can compile all the information known from the fossil record. That is the age, the time period that the fossil is found, and the taxonomic and or phylogenetic identification of those organisms. So what we're looking at here on the x-axis, we're starting in the Paleozoic, starting in the Cambrian, about 540 million years ago, and we're going to the present day. The Q refers to the Quaternary. Now, some geologists argue we are no longer in the Quaternary. We are in the Anthropocene. We are in a geologic age that is reflective of humans' impact on the planet. And we'll talk more about that in a later lecture. So what we can do if we have this information in the fossil record, going from one time period to the next, what we're looking at on the y-axis are the percentage of taxonomic families that go extinct. And by doing that, we can see that there are periods where there are large spikes of taxonomic families that are going extinct in the fossil record. And the two that we're going to talk about later in subsequent lectures are the end Permian extinction, which was likely the most dramatic extinction event experienced on Earth. And then the Cretaceous Paleogene, often called the KT for Cretaceous Tertiary event, where dinosaurs go extinct. Now, fossils also fill in the tree of life, gives us information about extinct groups, and gives us an idea of the sequence of evolutionary events. So we're showing you here in this phylogeny that major groups of vertebrates, the living lineages, are in bold. So here we have the amniotes with turtles, lizards, snakes, crocodilians, birds, and then up here, us, the mammals. Now what we see is that on the lineage leading up to mammals, there's a whole set of extinct lineages that are called the mammal-like reptiles or non-mammalian synapsids. Now, these non-mammalian synapsids are not a monophyletic group. They're paraphyletic. As you can see, we're including a common ancestor, but excluding all the descendant taxa, that is by excluding mammals, all right? So what do we learn about the evolution of mammals by knowing that these non-mammalian synapsids are more closely related to mammals than they are to other amniotes? Well, for one, remember in lecture we talked about one of the defining features of mammals is having 3 inner ear bones. And what we see is that 2 of these 3 inner ear bones are actually derived from bones of the lower jaw of these early diverging mammal-like reptiles. So we're showing you here in this diagram a early mammal-like reptile with an angular and quadrate. And we can see that as we move up the phylogeny, these different taxa, the angular and the quadrate become very small in size. And what we see in us, in the advanced mammal, is that two of those three inner ear bones are modified jaw bones that we see in the mammal-like reptiles. Okay, so in this brief overview of the fossil record, what I think you should take home, a take-home lesson of this, is think about fossils and phylogeny as the two pillars of macroevolution. You should roughly articulate the geologic timescale and understand that there's a huge amount of time, the history of Earth, that existed prior to the origin of life. You should think about how we use radioactive decay and isotopic ratios to date rocks, and then think broadly about what fossils can tell us about the history of life with regard to biotic assemblages, the rise and extinction of lineages, mass extinction events, and why filling in the tree of life with fossil taxa could really inform us about the origin of key traits in living organisms. So until next time, be well, be safe.